Welcome, good afternoon. I'm Thomas Lehner, and I want to welcome you to the Tuesday evening lecture of the Neurodegenerative Disease Working Group of the New York Genome Center. As always, quick housekeeping. If you have any questions during the seminar, please enter them in the chat box, and I'll read them during the discussion and at the end. Or you can also raise your hand and I'll ask you to speak then. So with that, it's a great pleasure to welcome Dr. Liam Holt as today's speaker. Dr. Holt is an associate professor in the Department of Biochemistry and Molecular Pharmacology at NYU's Institute of System Genetics. He and his lab study mechanical compression and molecular crowding and its effect on cell functioning in neurodegeneration, aging, and cancer. Please join me in welcoming Dr. Holt, whose talk is titled A Physical Model for Initiation of Neurodegeneration. Liam, if you could be so kind.
Okay, thanks very much. Okay, I'm going to share my screen. Okay, so as Thomas said, we're going to talk today about the physical biology of neurodegeneration. And I'm going to start for the first half or so of the talk with a conceptual framework that relies on previous work that, from the past couple of years. And at the second part of the talk, I'm going to go into really the title here and get into neurodegeneration. That second part of the talk is extremely new. And so be very excited to get feedback from you guys, but we don't have real answers for you yet. And a lot of the data are quite preliminary, but that's okay. You get a sense of where we're going. Okay, so the thing that we've been thinking about a lot is the biophysical properties of the interior of the cell, which are really quite extreme and unusual. So for example, here's a nice image from David Goodsell, fantastic structural biologist and artist. And what he's trying to convey here is just how packed with macromolecules, proteins and RNAs and so forth, the interior of the cytoplasm is. And it's actually about 40% of the volume of the cytoplasm can be taken up, can be excluded. And that's a huge volume fraction. So one consequence of this is quite intuitive, which is that things can slow down. And the cell is in fact thought to be close to what's called a jamming transition. And because we're in New York, I don't have to introduce that in any great detail. You've all been in jamming transitions of a sort when you've tried to go down FDR on a Friday. And it's kind of similar in the cell. If you reduce the amount of space available, you very quickly go from things moving fairly reasonably okay to like grinding to a halt. And these jamming transitions in real physical systems, they can occur at the kind of volume fractions that we see in the cell. So in a very simplified system with frictionless spheres, these transitions don't happen until 50 or 60% volume fractions. But our cells are not full of frictionless magical spheres, and we are not spherical chickens. So the molecules in the cell certainly interact with each other a lot. So for example, I have a colleague downtown at NYU, Jasna Brusic, and she's made these nice physical models of just synthetic spheres that stick to each other a little bit. And if you look at these, they're clearly forming network spanning, jamming clusters, and this is only a 20% volume fraction. So the cell could easily grind to a halt. But this depends a lot on what we call length scale, which is simply how big things are. So again, if you've been in New York City on Friday at 5 o'clock, you don't want to be in a car or a truck, you want to be on a bicycle, because the smaller things can move between the large things that are jammed. And the same is true in the cell. So the length scales that we see in the cell can span many orders of magnitude. And so, you know, the crowding, macromolecular crowding that I'm going to talk about doesn't really matter for a potassium ion, but it matters a lot for a vesicle. And this was really driven home in an important study by Christine Jacobs-Wagner about 7 years ago now. And here she demonstrated that, in this case, in a bacterial cell, what I'm going to show you is this synthetic particle in yellow, and it's inside E. coli here. And we're looking at a movie, and this is time in seconds, I think. And you can see this particle just bouncing around in a normal cell. But on the right, what she's done is she's poisoned the cell and depleted ATP. And what you see is that this particle just stops moving. And what the conclusion was from this paper was that you need active processes inside the cell to keep things at a certain size to allow them to move. And again, this goes back to length scale. So if you look at small proteins like a GFP molecule, it moves similarly with or without ATP present in the cell. But this particle, which is about 100 nanometers in diameter, it really relies upon active processes for the cell to not undergo a glass transition, which is a particular type of jamming transition, and everything to grind to a halt. So the motion of particles inside a cell, I'm going to come back to this lots and lots of times. The motion of particles in a cell depends on how big the particle it is. It depends on how crowded the interior of the cell is. And it can also be modified by ATP consumption. And you can imagine ATP driving motors that stir things up in the cell. Okay, so that's in an unperturbed cell. But how about if we start to squeeze on cells? So this is one of the things that we study, so compression. So this is a microfluidic device that we've used in the past. And here we're putting cells, in this case, very simple cells, yeast cells, into this chamber. And this chamber is designed such that as the cells grow, it's made of a material that's elastic, and so it will be deformed. And this place where we load the cells will pinch and completely confine the chamber. I want you to also pay attention to the shape of this chamber, because the distortion of the shape tells us how much pressure is building up in the system. So here you see the loading channel pinching off and the cells building up pressure. And because of that, they're pushing the chamber to be wider and wider. And by the degree of distortion, because we know the elastic modulus of this material, we can calculate how much pressure is in this chamber and how much pressure on average a cell is experiencing. Now we can look at a molecule moving inside these cells. This is actually an mRNA molecule moving around. And at relatively low pressures, these molecules move okay. And here at somewhat higher pressures, the molecules, their motion has really decreased a lot. And we have a bioRxiv preprint about this subject up right now. And one thing that we observe is that the growth rate of cells, again, these are Cerevici cells, decreases exponentially as a function of pressure. And something similar is seen with mammalian cells as well. Okay, so what does that tell us? That tells us that the cell is very crowded, and if you push it, and all our analysis suggests that if you push it to be a bit more crowded, then this crowding becomes limiting for growth and cellular processes. And so this is interesting because if you look across all domains of life, be it a bacterial cell inside a virus, at the synapse of a neuron, crowding is very similar. It seems to be a quite conserved property. About 30 to 40% volume is excluded. It doesn't have to be that way. We could have the cell at potentially 20% crowding, and then compressing it a bit wouldn't make so much difference. It would have room to move. So, going back to this important study by Jacob Peter Gowie on the melting temperature of wax, why are we flying so close to the sun? It kind of implies to me that there might be some benefit to crowding as well. And so people have thought about this a lot, particularly Alan Minton has written a lot about this. And here's one potential benefit. So if you remember back to your physical chemistry, and just consider two molecules that can bind together or they can be separate. So there's a binding reaction. If we plot the rate of some reaction that depends on these two bound molecules, this heterodimer, let's think about what determines whether the heterodimer is formed or not. So it depends on the positive sort of enthalpy, the advantageous binding energy of these two things coming together. But there's also this problem that entropy is going down. And the universe doesn't like that. In general, you want to have entropy always going up. So this term, which determines whether the reaction goes this way or not, is strongly influenced by the fact that these two things coming together reduces entropy. But we can offset this effect if we simply add a crowding agent to the system. Because since we're taking up lots of space with this crowding agent, these two things that are apart also take up space. And when they're apart, they take up more space than when they're together, such that the binding of these two things actually creates space and degrees of freedom for the crowder. Thus, the crowder's entropy goes up. So this unfavorable term for entropy for binding of the things we care about is compensated by the favorable entropy term for the crowder. What this means altogether is that as you initially increase the crowding concentration, the rate reaction in the cell starts to go up because of this entropic effect. But then at a higher concentration of crowding, things start to get too viscous and they jam, and the reaction rate goes down again. And anyone who's done a DNA ligation reaction and then compared that to a rapid DNA ligation reaction, so one takes an hour to overnight, and the rapid ligase kit takes 5 minutes or 10 minutes. The difference is that the rapid ligase kit has crowding agent in it. So, in your own biochemical experience, you may have come across these effects. So all of this suggests that there should be some optimal level of crowding inside a cell. And I think that that's why this 30–40% crowding value is so conserved across biology. Okay, so I'm going to tell you now about the initial studies that set up this project, which are driven initially by a graduate student, Greg Brittingham, and Morgan Delarue, who now has his own group in Toulouse. So what are the mechanisms that control the degree of crowding in the cell? So first of all, how do we look inside a cell and get an idea of the physical properties of the cell interior? So what we're going to do is we're going to rely on this technique called microreology, which is, we can't just look at a transparent cell and know the material properties of that cell. But what we can do, if we can see something fluorescent inside the cell, we can infer those properties. So imagine you have this green dot inside some material that you can't see. By watching it jiggle around, you could guess that this environment was quite crowded and this environment was less crowded because the motion of this green particle is less constrained. So that's a gross simplification of the field of microreology. But there's a whole physical framework to get lots of different information out from this. And I'll go into a bit more depth as I go through the talk. So this has been taken advantage of over many decades. Francis Crick did this back in the 50s. There was a major study by Kathy Luby Phelps in the 80s. And what people have done quite a bit in the past is introduce non-organic, so polystyrene beads or quantum dots or magnetic particles into cells by things like microinjection. And that's great, that's worked, but it has a certain number of limitations. So a major, perhaps the major limitation is that it's very labor intensive and very low throughput. But additionally, you can also disturb the cell and dilute the cytoplasm by doing these kind of procedures. And it's not possible in lots of contexts. For example, in organisms with a cell wall, or if you're embedded in some tissue and it's really hard to get the needle in there. Okay, so being pragmatists, we wanted to come up with a much easier solution to being able to get these passive nanoscale probes into the cell. And what we came up with is what we call genetically encoded multimeric nanoparticles, or GEMs for short. So these are based on a gene that we introduce into a cell. And the gene has two parts. It has a scaffold, and it's got a fluorescent protein, GFP. So the scaffold is, so this is a monomer. And so here's the scaffold protein and here's the GFP, but the scaffold self-assembles in many copies into a icosahedral, perfectly defined structure with a known diameter. And this is a homomultimer. So one gene, you get a bunch of monomers, and all of these things assemble automatically within a cell. And the size of this thing, this 40 nanometer diameter, we call this the mesoscale, which is, you know, the in-between scale. So there's the nanoscale, which is the scale of individual protein molecules like GFP. Then there's the micron scale, which is the scale of things like organelles, like the nucleus. And then there's this very important in-between scale, the mesoscale, which is the size of things like ribosomes, polysomes, vesicles, you know, all of the major control molecules in the cell, like RNA polymerase, DNA polymerase, proteasomes, you name it. So it's very important. And it's also a very interesting physical position, because if you remember from a few slides ago, in the study by Christine Jacobs-Wagner, what she found was that in the nanoscale, this active component of the cell didn't matter. But as soon as you hit about 50 nanometers or so, this mesoscale, then the cell becomes glassy and requires active processes to stay fluid. Okay, so here we are expressing our GEM nanoparticles, and this is in a pancreatic cell. And this is real time. So we have hundreds and hundreds of particles. This movie is looping every 4 seconds. We're imaging very quickly, so 100 frames a second. And what we're doing is we're directly detecting these particles in every frame and tracking them. So here's the same thing in a yeast cell. We like yeast because we can do really quick genetic experiments. And so here's the tracking in the bottom right here. So we're taking these tracks, and from these tracks, we can do all these biophysical analyses. All right, so what did we find? So something we found very quickly was that depending on the metabolic state of cells, the global behavior of these nanoparticles changed. So we kind of stumbled across this by accident. We found that when cells, and this is true of everything I'm going to tell you right now for the next few slides, we did in parallel in yeast cells and in human cells, and the results were very similar. Yeast were great because we could do screens and figure things out quickly, and then validate them and find what was the same and different in human cells. And in this case, pretty much everything was the same in yeast and human. So what we found was that in amino acid starvation conditions, the diffusivity, the effective diffusion of these nanoparticles increased. And that could be due to one of two possibilities. So the motion of these nanoparticles is affected by how crowded the environment is. It's also affected by how active the environment is. So I'm not going to go through the evidence for this, but just take my word for it for now. I can come back to it at the end. It was because of crowding in this case. So what I'm going to tell you is the mechanism that we think we figured out so far, some consequences of that, and then I'm going to move on to neurons. Okay, so amino acids are the metabolic condition that changed the global behavior of these nanoparticles. And so, of course, we immediately thought of the TOR kinase, target of rapamycin, because this is the major sensor of amino acids in cells. So TOR, it integrates information about growth factors, amino acids, etc., and of course, stress. And it controls, well, it controls everything, which is great, but it's also kind of annoying. But this is why it was good that we had yeast. So TOR is also very handy because, of course, the name of target of rapamycin comes from the small molecule that was discovered on Easter Island, otherwise known as Rapa Nui. So this compound rapamycin will very specifically turn off this kinase. So we can do a simple experiment to ask if TOR kinase is the thing responsible for the change in behavior. We just add rapamycin. So this is what we're doing. These are yeast cells. So this is control condition. We're adding rapamycin, and what we see is that the motion of these particles accelerates. And we can quantify that here. And it's very, very reproducible. We've done this experiment probably 1 million times now. Okay, so the next question is, well, how does this happen? Why are the nanoparticles going more quickly? I already told you that we think it's because crowding is changing, but what's the mechanism? So again, TOR controls everything. So this was super annoying because we had a bunch of hypotheses that in retrospect were probably silly. And we tested 100 different things, literally, before we finally came to what we're looking for. So what we were looking for was a mutant where this effect that when we add rapamycin, the particle diffusion goes up. We wanted this effect to go away. And we found that mutant, and several mutants in the end. But the first one that was super striking was this gene SFP1. And what you can see actually is that even in the control conditions without rapamycin, the nanoparticles are moving quickly, and then adding rapamycin has no further effect. So what is SFP1? SFP1 is the major transcription factor downstream of TOR that controls the rate of ribosome biogenesis. So of course, when TOR kinase is active, it drives cell growth. So you need lots of ribosomes. And when the cells are stressed out and TOR is inactive, ribosome biogenesis turns off. If you delete this gene, SFP1, ribosome biogenesis, I mean, it's still happening, but at a very low rate, at the basal rate. So we looked through a few more mutants, and we found that the mutants that had a similar behavior all fell in these two classes. One is ribosome biogenesis, and the other one was autophagy, which again is controlled by TOR. And again, autophagy, it controls the degradation of ribosomes. So these two processes set ribosome concentration. And seeing is believing, so we collaborated with my friend Ben Engel, and we did this cryo-EM tomography of the cells. And so what I'm going to show you is this 3D reconstruction. And in cyan, these are ribosomes. And this is why we should have thought of this as a mechanism from the very beginning, because it turns out that ribosomes are the most space-abundant thing in the cell. So in terms of absolute number, they're not the most abundant things. But in terms of the space taken up in the cell, ribosomes account for half of the volume taken up by macromolecules in the cytoplasm. They're massively abundant. And if you inhibit TOR, what you see is that that concentration of ribosomes goes down twofold. And we came up with physical models, and we could explain pretty much all of the changes in the motion of the nanoparticles based on ribosome concentration. Okay, so cool. So TOR is having a major effect on the physical properties of the cell interior, the cytoplasm interior, based on ribosome concentration. But who cares? Are there actually any consequences of these crowding changes? Well, so crowding can do two things, as I said before. It can change the way that molecules move, and it can change the way that molecules assemble. Remember back to that those molecules binding one another, the entropic effects can drive molecules to bind together. Crowding almost pushes molecules together. And remember that when we get to the neurodegeneration side of things, we haven't got that yet, but one of our major hypotheses is that if crowding gets too high, you might be favoring assembly of molecules too much, and this could lead to aggregation, for example. Okay, but I'm getting ahead of myself. So first of all, what we looked at was something that we could readily visualize, which is this process of phase separation. And this is one of those things where, you know, a couple of years ago, I might have asked who's thought about this, and most people would say no, but now I think in cell biology, at least, it's become a very hot topic. And we're currently in the Ralph Wiggins stage of the field where people are happy and angry. Nevertheless, this is an important biological phenomenon, whether it's being overinterpreted to some degree in some places or not. But the concept is simply that if you have multivalent molecules in the cell, they can, at a certain concentration and in certain conditions, start to favor association with one another more than they favor association with the surrounding solvent, and can spontaneously demix, like oil and vinegar, into large assemblies. And this was, a field revitalized in 2009 by Cliff Brangwyn and Tony Hyman, and has taken off from there. Of course, people have thought about this for 100 years, but this is the paradigm that we chose to look at, because as I said, multivalent molecules can assemble into large assemblies. So things like the fuse protein that's involved in ALS do this, TDP43 can do this, et cetera, et cetera. One very convenient thing about these large condensed forms of these molecules is that you can visualize them quite easily in cells. And something that people have perhaps not spoken about that much is that this process is enthalpically quite favorable sometimes, but entropically quite unfavorable, because you're demixing. You're going from a well-mixed system to a demixed system. But again, adding a Crowder here can really favor this demixing, because the Crowder gets way more space available to it when the condensate condenses. So crowding can drive phase separation. And this is, of course, physically and theoretically true, but is it the case for the specific biomolecules that we're interested in, i.e. ribosomes in the cell? So to ask this question, we turn to a synthetic system, because if we looked at any real endogenous phase separating system, you always have the problem that if you're messing with TOR kinase, which phosphorylates tens of thousands of things, then how do you know you're not looking at regulation? So we wanted to look at something that was relatively biologically inert, but does phase separate. So we chose to use the system developed by Mike Rosen, which are the sumo-sim systems. So it's a synthetic concatemer of multiple sumo domains that will bind to a concatemer of sumo interacting motifs. So the orange thing interacts with the blue thing, and you can form networks. And these networks form little droplets. And so this is an experiment completely in vitro. And we know everything that's in the test tube. We've got some of the blue things, some of the orange things, and will they form droplets or not? That's plotted on the y-axis, which is the partition coefficient, which is the concentration in the solute versus inside the droplet. And on the x-axis, I'm just varying ribosome concentration. And I'm using ribosome concentrations that are exactly the ribosome concentrations we find either in a rapidly growing cell, which takes up about half of the volume of the cytoplasm, or in a cell that's treated with rapamycin. And you can see over this physiological range of ribosome concentrations, you can have a pretty substantial effect on how much droplet formation is happening in a test tube. And of course, this is massively simplified, but at least we know what's going on. The cool thing is we can also do this in a cell. So we can express this, an M-Cherry tagged version of this sumo-sim system, and it will form large droplets in a yeast cell or in a human cell. And then we can treat the cell with rapamycin, and what we see is that these droplets either completely dissolve or they get much smaller. We can quantify that here. So we have the total area of droplets in the cells, treat with rapamycin, it goes down substantially. It's actually a much bigger effect in cells than we see in vitro. So there's interesting things going on there. And we can recompress cells. So we can use sorbitol to osmotically reduce the volume of the cell to recrowd them. And then we recover a good deal of the droplets. So we get this idea now that tuning molecular crowding in the cell tunes molecular assembly. And here we're looking at a synthetic phase separation system, but you can extrapolate that probably to many, many reactions and many types of molecular assembly in the cell. Okay, so that's setting the whole framework for the hypothesis. Molecular crowding is important. It has a big effect on molecular motion, especially for things that are in the mesoscale or larger. So, 30, 40, 50 nanometers or above. But it also drives molecular assembly. So it affects the kinetics of assembly and disassembly of multi-macromolecular complexes. So that's crowding. The other thing that affects motion in the cell is active processes. And so if you deplete ATP, things can stop moving altogether. So remember that activity is important too. And in this first part, I've told you our research that TOR kinase tunes crowding through ribosome concentration in the cytoplasm. And I can tell you about the nucleus if you ask me at the end. All right. So that was a long introduction, but let's get into a hypothesis for the initiation of neurodegeneration. And this has been made possible by a seed grant from the Chan Zuckerberg Initiative, the Neurodegenerative Challenge Network, and this was with Hamali. And so that was a fantastic opportunity. And it gave us the opportunity to then expand this into a larger collaboration that's been funded through the Transformative Research Award with Hamali again, but with the extra participation of fantastic additional collaborators, Ai Yamamoto, also at Columbia, and Jenna Gregory in Edinburgh. And there are numerous scientists that have been working so far on the project. So Risa Kimura in Ai and Hamali's lab, Gane in Hamali's lab, and then in my lab, we have Martina Benucci, who's really been trailblazing this. And she's been joined by Yishuan Seng more recently, and Tong Xu has been doing some of the physical analysis. So I'll repeat those names as I go through. So what is this hypothesis? So the hypothesis then just builds off of what I've been telling you so far. Changes in what we guess, or what we would like to investigate, is the possibility that changes in the biophysical properties of the cell interior as a function of age play a role in the initiation of neurodegeneration. So of course, we don't know what drives the initiation of neurodegeneration, period. And especially the vast majority of neurodegenerative disease is spontaneous. It doesn't have an obvious genetic underpinning. So what's happening? And so we don't claim that this is necessarily the entire story, but we'd like to investigate the possibility that the cell environment itself is an important factor. And we'll see if we're right or not. So this could be changes in crowding inside the cell. And why would the crowding change inside the cell? So this could be things like changes in metabolism as a function of age. We know that if you give animals rapamycin, they live longer, for example. It could also be things like changes in tissue structure. And then the other thing is we should always keep an eye on this other side of the coin, the active processes, the ATP-driven motions in the cell. And oh, I forgot to mention in the previous slide, also, we've been thinking a little bit about vascular defects as another possibility. And so again, these changes are going to be essential for molecular motion and assembly if they occur. And that's eventually what we'd like to look at as well. All right, so far I've told you about very simple model cells, and we haven't been talking about neurons at all. And of course, neurons are very different than HEC293 cells, and they're definitely quite different than Saccharomyces cerevisiae. So this is illustrated by this really nice cryo-EM from this year from the Kulkulski lab. And what we're looking at here is we're zooming into the interior of an axon. And there are a few notable things here. So in blue is the plasma membrane, and purple, we're going to come back to this, microtubules. There's massively dense microtubule arrays going down the axons and all neurites. The endoplasmic reticulum is super important in cyan here, and that's something that Yishuan is starting to think about. Mitochondria are going up and down, and vesicles are going up and down. There aren't actually that many ribosomes, so that's very different. So, it's a very different physical system, and just looking at it, just getting a sense of what that looks like at all in a material sense is something that there's very little understood. So we're very excited just to get an initial survey of what's going on here. So this is kind of like a very big framework for the Transformative Research Award grant. All I'm going to talk to you about today is these cellular models, but ultimately, I's group is going to work with mouse models and make really cool ways of perturbing things like autophagy and the osmotic conditions inside the brain. Of course, Jenna has got amazing tools for looking at human pathology samples and looking at things like TDP43. And Hamali has amazing technologies for doing things like spatial transcriptomics and proteomics. And we hope to integrate all of these things and cycle back and forth to get a sense of whether any of this stuff is relevant to neurodegeneration. But today we're just going to talk about GEMs. Okay, so the first thing is, do they work? And of course, this would fall pretty flat on its face if that answer was no. So Martina did this experiment, and we were very relieved when we found that we could get these nanoparticles. We introduced them into, these are human-induced pluripotent stem cells that have been differentiated into different types of neural cell. And we, Martina, transduced them with lentivirus that encode the GEM gene. And now these cells, this is a real-time video, and you can see a bunch of cool things already just from looking at this. The particles are there, they're moving around, they're getting into these neurites. And we know from the work with Martina and Gane that they're getting into axons. We've marked axons. So that wasn't a given, so we can analyze the physical properties of these different compartments at a subcellular resolution. Yishuan has been thinking about using a different cell system with Esteban Mazzoni. And so we're going to have like various different types of neuron, and that's going to be really exciting. But I'm going to focus on the work that Martina and Gane have been doing initially. And they immediately found something really cool and unexpected, which is that if you compare the diffusivity of GEMs in the soma versus in neurites, you don't see what you expect. So here's a chymograph. So this is time on the y-axis, and this is the x is the position of GEMs, just to show you if you're familiar with this kind of representation. But I mean, it's just the motion here. So what I'm showing you here is the y-axis is the rate of motion, the diffusivity of the GEMs in these two compartments. And what we found is that the GEMs are moving more quickly in the neurites than in the soma. And that's not necessarily what we expected from the simplest possible expectation. So the simplest possible model is the kind of thing that Tong has been trying to develop a framework for, where she's been doing these molecular dynamics simulations of crowded environments, and just figuring out what our ground expectations are for a very, very simple colloidal system. And what you expect is that simply due to geometry effects, if both the soma, which is in this case a cube, and the neurite, which is in this case an elongated rectangle or cuboid, is everything's the same except for the shape. If that's the case, you'd expect that the particles would move more slowly in the neurites. And of course, I already told you the environment inside a neurite is very, very different than inside a cell or the rest of a cell. So perhaps we shouldn't be too surprised, but it's cool. And we can start to think about, well, why is this? So there are a couple of possibilities. So it could be that the neurites are less crowded than the soma, or it could be that they are more active. There's more like ATP burning activity that's increasing the effective temperature inside there. Or it could be a combination of these two things. So one thing that we can do, just going beyond the very simplest idea of what we can do with particle tracking, is look at angle frequencies when we look at these tracks. So if we just consider this green particle taking one step and then a second step, and the black arrow and then the magenta arrow are step one, step two, what you can do is you can ask, what is the angle that you get from one step to the next? And so just to get your mind around this, what we do is we take this black line and this magenta line and we make a graph out of them. And we take the angle right here, and it's about 45 degrees. So in a completely diffusive Brownian system, these angles should be totally random. It should be equally probable that from here to here, you would go here or here or here or here. Like any direction on the second step should be equally probable. Now, let's say you see that you start here, you go one step, and then your second step is almost completely backwards. So now we take these two lines again, the black and the purple, and we put them next to each other, and we get this angle, which is close to 180 degrees. So this is, if we see more of this kind of angle than expected, that's what you get in very crowded environments. Because, for example, you crash into some crowder, for example, a ribosome, you bounce off it and you go backwards. So if you tend to always go backwards, then that indicates crowded environment. Another possibility is that you start here, you go to the second point, and then the third point. If you put these two, the black and the magenta arrows next to each other, the angle is close to 0. And that's what you get if you have a quantum soccer player getting inside your cell and kicking the thing in a particular direction. So that's indicative of active directed motion. Now, active random motion wouldn't do anything. The diffusion coefficient would go up, but you wouldn't see any change in angle distribution. So it needs to be both active and it needs to be directed. Okay, so what do we see inside neurites? So what I'm showing you here is on the y-axis, the frequency of different angles. On the x-axis, the angle, going from 0 to 180 degrees. And what you first, and in blue, I'm showing you the distribution for neurites. In red, the distribution inside soma. And the gray dotted line is what you expect random. So the random expectation is that everything should be the same. So what you see in both neurites and in soma is that this 180-degree angle is very obvious. So that it's crowded, and this is expected. We've seen this before. What's perhaps a bit less expected is that this close to 0-degree angle is more frequent than expected by chance only in the neurites. So it implies that there's some active directed thing happening inside neurites. And that's really cool, and something that we weren't expecting again. Something I should say is that the speed of the analysis is, we're looking at the motions of things that are like ribosomes or, macromolecular complexes. They move really quickly. So these time scales that we're thinking about are in milliseconds, right? It's very different than, fast axonal transport of vesicles down axons is actually much, much, much slower than what we're thinking about here. So I just wanted to give you a sense of time scale. So, okay, what might be going on? So one thing that I want to come back to is the fact that the interior of an axon is full of microtubules. So here's a bioarchive paper from Lucas Capitan. And what they've done here is they've done some expansion microscopy. And what he's showing is the distribution of either acetylated or tyrosinate microtubules throughout the axon. And what you see is this incredible density. The most stable, what are predicted to be most stable microtubules are in the center, and more active, dynamic ones are predicted to be on the outside. So that's really cool. So we thought, well, perhaps microtubules are going to be important. They're depolymerizing and polymerizing very quickly on relevant timescales. And so, and this is something we can test because, what microtubules do is that they polymerize, and then there's dynamic instability. So they have a catastrophe, they depolymerize, and then they get rescued, and then they're growing and shrinking all the time. And this is massively regulated by endogenous proteins, including things that are very important for neurodegeneration, such as tau. And we also have pharmacological agents that we can use, such as taxanes, like paclitaxel, which stabilize microtubules. And we have, so this should say destabilizers. So we have things like vincristin, which will depolymerize microtubules. And you can imagine that a couple of things would happen. One is that if you stabilize microtubules, they might be more dense in there, but they're also less dynamic. And if you destabilize them, they'll be less dense. And so what we did, so this is Martina's results. So we're looking now at the diffusion of the gems in the neurites. And there are several different drugs here. And what you see is that this drug, which inhibits dyneins doesn't make too much difference. But stabilizing microtubules with paclitaxel decreases the diffusivity of the gems, whereas destabilizing it increases the diffusivity of the gems. So that's a very interesting indication that microtubules are an important component of what we're seeing. We have a sort of analogy here to a recent paper, a collaboration we did. So this is Lehner Carlini's work in Tarun Kapoor's lab. And when I say collaboration, I mean, we talked a bunch and Lehner did all of the work. So what she did is she looked at how these same nanoparticles moved inside the mitotic spindle, which is a similarly dense array of microtubules. In fact, inside the mitotic spindle, microtubules are so dense, there's only about 20 nanometers on average, between each microtubule. And remember, gems are 40 nanometers in diameter, so they shouldn't even really be able to fit in here. And yet, in Lehner's data, so what I'm showing you here is she tracked the nanoparticles in the cell outside of the spindle zone and inside the spindle zone, and did this for many cells and got thousands of tracks. And this color coding here is based on the diffusion coefficient of the nanoparticles. And what you see in this representation here is nothing. i.e., there isn't this really slow spindle zone or excluded spindle zone and then a fast cytoplasm around it. Everything's the same. And it turns out that the dynamic instability of the microtubules inside the spindle are crucial to essentially normalize the rate of motion of particles the size of gems, which would include things like, the gamma tubulin ring complex. So the cell becomes normalized biophysically because of the motion of microtubules. So I think it's really interesting to consider that this could be a really important thing when you start evolving a neuron now. Okay, so that's microtubules. Martina also wondered about actin, because there's also extremely organized actomyosin and actin spectrin rings all around axons that help determine the diameter of these structures. And so she did an experiment with blebbistatin, and this is very preliminary, but we see that with vincristine, which is the microtubule depolymerizer, which we think creates more space, diffusion goes up. And also with blebbistatin, which has been showed, this is a myosin inhibitor. It's been shown that this increases axon diameter, so potentially gives more space. That also increases diffusivity. A quick aside, so these effects are very dependent on culture conditions. So what we found, like Martina's original experiments with Gane were in microfluidic devices, and later we moved to go a bit more quickly into simpler systems, and we found that the density of cells was very important. So at low density cultures, this difference between the neurites and the soma was not evident. But once you get high density cultures, you can see the soma are all clustered together. We think that that's when the soma diffusion goes down. Actually, the diffusion inside soma is quite a bit lower than in many cell types we look at. And so we sort of speculate that maybe the soma are all squeezing together into these clumps, and that's reducing the cell volume and increasing crowding. This makes us think about how cells respond to changes in tissues as well. So tissues are changing substantially as we age. So a friend of Hamali and mine, Kevin Shalou, had an amazing paper a couple of years ago. And one of the amazing things that he found was that the stiffness of the ECM in mice, at least, changes radically as a function of age. So one thing that we wonder about is whether these mechanical properties to tissue could play a role in changing, in being propagated forward into changes in physical properties in the cells. So that's a hypothesis we want to test. One thing that we've been doing in that direction is using a very simple system that was developed by Greg Brittingham. So he developed this 3D printed wafer embedded in agarose that you can put on top of cells and apply a compressive stress that way. And we use this extremely high-tech device, which is a washer from a hardware store that we can stick on top, and different amounts of these washers will give different amounts of compressive stress. And this is very preliminarily, but we found something actually extremely surprising if this turns out to be true, which is that in the soma, compressing quite a lot actually didn't make too much difference, but the neurites, they were affected. I kind of would have expected naively the opposite, but I'm naive, so there you go. We'll see if that holds up. Okay, so I'm wrapping up. Just to point out that we have extended this nanoparticle technology. We've directed them to the nucleus here on the left. Whoops, didn't loop this movie. So this is the cytoplasm, the nucleus, and we've even put them in the endoplasmic reticulum. So this is something that we're very excited about because we think the ER could be very important in certain types of neurodegenerative condition. All right, so here's the conclusions from our preliminary report. GEMs are extremely powerful tools to characterize the physical properties of the interior of neural cells. We've been focusing on differentiation protocols that will give motor neurons, but of course, we're very curious if different types of neuron have different properties, and then other neural cells like, glial cells, for example. What we found so far is that diffusivity is higher in neurites than in the soma. And that part about the soma depends on the culture density. So microtubule dynamics might be a part of this. They seem to be driving some directed molecular motion within neurites. And finally, that neurites seem to be more sensitive to mechanical compression than the soma. So going forward from this, ultimately we want to see whether some of these effects that we're seeing about molecular dynamics and molecular assembly are relevant to proteins that have been implicated in neurodegenerative disease. But certainly so far, we're seeing some interesting things from a biophysical property of the cell perspective. So I'm going to wrap up right there. Just to reiterate all of the thanks, Martina and Yishuan and Tong from my lab have been doing an amazing job. Risa, Ganei from Hamali and I's lab. And then, of course, amazing collaborations and discussions with Hamali, I, and Jenna throughout all of this. This is the rest of the people in my lab, and want to give a shout out to all the funding sources. We have various other projects beyond science and science communication. If you want to find out about those, let me know. And we have positions available. So if you know of anyone looking, let us know, or just scan this thing and give us feedback. So I'll finish there. Thanks a lot.
Thank you, Liam. That was wonderful. Questions for Liam? So Liam, could these biophysical properties then be considered as risk factors, as vulnerability that for neurodegenerative disorders that have other causes?
Right. So that's part of our hypothesis that it would be very interesting to compare cells from different individuals with no genetic predisposition to disease, but that did get disease, and see if there is a biophysical difference. And that's one of the things we have planned. So there are a couple of hints out there that certain tissue scale things that we think would change the physical property of the cells correlate with neurodegeneration. For example, edema and problems with vasculature seem to be coincident with sites of aggregation. And so, osmotic compression is a very powerful thing that will massively change the cell properties. So that's one idea we have. I'm going to allow Hamali to answer any of these questions as well, because she knows about 10 times as much as I do about any of these things.
Liam, it's interesting, I think, that if you look at neurons with ALS-related aggregates, the aggregates are always in the soma. And I wonder if in those cases, the movement in the neurites would be the same as you see in these normal cells. That is to say, could these protein aggregates associated with neurodegenerative disease actually really affect what's going on with crowding in the neurites?
Yeah, that's a great question. I mean, we need to find a model system where we can induce those TDP43 aggregates. So that's something that we're working on. And we'll just have to do the experiment and see. One thing that's really interesting about TDP43 that Martina's been thinking about is its role in splicing, and the fact that Stathmin is, lost, its expression is reduced in Hamali's work and other people's work. And Stathmin is involved in microtubule stability. So, we see that there is a link between microtubule stability and neurite diffusivity. And so maybe that could be an indirect way in which TDP43 could play a role in the neurites.
Well, and TDP43 movement in neurites is changed as well. And so that could also be related to that.
Right. Right. So we often think about these large aggregates that are found in pathology, but we're very, very interested in much smaller scale, faster and smaller things happening, so that are invisible to pathology. But if we use very high resolution imaging, we might be able to start to get a handle on, for example, assemblies of tens of molecules instead of millions of molecules, and how those dynamics could be affected as well.
Yeah.
So we have two questions, related questions in the chat, but from Larry Chassin and Alison Goat that I will try to combine. So, do mapped mutations influence gem movement, and can you manipulate gem movement using microtubule states using drugs?
So the drugs question, yes. So those were some of the data that I showed from Martina. They're preliminary, but we do have quite a lot of pharmacological agents that we can use to ask that question in detail. So the microtubule stabilizing taxanes reduce the diffusivity of gems, and the microtubule depolymerizing agents increase the diffusivity. In the former case, there are still two possibilities for this. One is that the dynamics are reduced, and that is the reason. The other is that the crowding has changed, and we can dissect those. We just need to go deeper into the analysis. The question from Allison about mapped mutations. I'm going to ask Hamali about that one.
Those are mutations in tau. We haven't tested that yet, but those are exactly the kinds of things that we want to do.
Yeah, so tau is super interesting, of course, because of its role in controlling microtubule organization. And so one could easily imagine that there's some feedback there. I think a lot about the assembly of proteins at different scales. And one thing that's been suggested is that tau condenses into these large structures. I'm not sure how well accepted that is. But whenever you're trying to condense from something that's a few nanometers to hundreds of nanometers, you can become what's physicists called frustrated if the environment is too crowded. So you aren't able to make the full size structure just because you can't move individual sub-assemblies together. So tau would be interesting in that context.
So we have a question from Larry Chassin. Larry, you can talk now.
He's muted.
I'm unmuted now.
Yeah, you're unmuted, yes.
Yes. I wonder if you can use the system and look at electrophysiology at the same time.
Oh, yeah. Yeah, I think we can. Yeah, as long as we can stick a needle in there, we can definitely do that. And, we can either do electrophysiology, or we could pharmacologically do something simple at first and just, drive, hyperactivity or inhibit activity and see if we see something there first. But then, really see if we can get strong spatiotemporal correlation between, action potentials and changes in the physical. That would be super awesome. Yeah.
Simon has an interesting question in the chat too.
Okay, well, we'll get to Simon. Louis Rackert has his hand raised. Louis, you can talk now.
Yeah, first, I thought it was a very interesting talk. I was just wondering if you could tell us a bit about the approaches you're thinking of for looking at postmortem material.
Ooh, I'm going to defer to Hamali on that one as well. To better details.
So one, we need observations that, so we obviously can't put gens into those systems, right? So we need correlative measures that can span both the in vitro system and the postmortem system, and also the mouse systems. So one of those things is looking at smaller assemblies of things like TDP43 instead of these larger antibody-based things that only detect very large-sized aggregates. So one of the groups at Edinburgh that's also involved in this project has developed aptamers that recognize different conformations of TDP43 and can pick up assemblies ranging in size. And because this is an extensively characterized cohort, both clinically and pathologically, we can begin to relate using these aptamers where we see these aggregates or proto-aggregates. So we can see them in both the soma and in the neurites, and we can begin to relate these to other molecular measurements or correlates that we can measure from the other systems that we're using. So this is part of the reason why we're integrating these studies across these different models and different scales. With the postmortem systems, we can also begin to look at changes near the vasculature, see where there are areas of clearing. Are there foci where you can see seeding of these assemblies and the beginnings of compression, and where you begin to see foci for gliosis and things like that. So those are the hints that we have from the postmortem system. And these are some of the tools that we're hoping to apply to bridge the observations on the in vitro system and on the in vivo systems.
Okay, thank you.
Question from Simon John. How would you predict stiffer versus more flexible cells? ECM would affect the response of gem movement in response to pressure. Do you know what levels of pressure difference are needed to make a difference?
Okay, so there's sort of two questions embedded in there. So maybe I'll take the second one first. So we've done a lot of work with compression of cancer cells. So that's another aspect of what we do in the lab. And so, the kind of pathological pressures that build up inside of tumors, they're not huge, but we've been mimicking them, and they have a huge impact on crowding in cells. So we're talking about hundreds of pascals, and yeah, I can easily imagine those kind of things, those kind of pressures building up in a perturbed tissue. So those are the levels of pressure needed to make a difference, hundreds of pascals, which is not much. So how do you think stiffer versus more flexible ECM would affect things? So we know that the stiffness of the substrate on which cells are growing impacts their volume. So this, relates to a really interesting body of work that there are lots of points that have not quite been satisfyingly connected, but they start out with work in stem cells showing that in different stiffnesses, you get different differentiation. Dennis Disher showed that in 2006. And then more recently, Dave Bates and Ming Guo and Jennifer Lippincott-Schwartz showed that if you put cells on different stiffnesses of substrate, one of the things that really changes is the volume of the cell, which is maybe not super intuitive, but you can imagine if the cell is kind of pulling down on its substrate, then the more it can pull, the more it sort of hunkers down and water gets forced out and its volume goes down. That's what happens to mammalian cells, at least mesenchymal stem cells. People haven't done that as much with neural cells, to my knowledge. It's one of the things that Martina is starting to look into. And it's definitely one of the questions we want to think about. This is a major thing we want to look at. So we're starting to set up the ideal models for synthetic extracellular matrix, because you actually have to be very careful about how you design those experiments. For example, if you take collagen as your simple ECM and you just change the concentration of collagen to change the stiffness, that's actually not a controlled experiment, because you also change the concentration of ligands that the cell binds to and signals from. So you need some special technologies to do that. That's one of the things that Kevin Chalou has been generating. So, and then how does stiffness and pressure relate to one another? Yeah, that's another really interesting question. That would be the final piece of that puzzle. So we plan on looking at all of those things.
And thank you. And the final comment is from Alison Goat to, we have isogenic lines with tau mutations. Some mutations change splicing, but not protein sequence. Might be interesting to see if they affect gem movement.
Okay, interesting. Yeah, that would be great. We're always definitely looking for opportunities to survey different types of cell. We have lentivirus, and so it's not a crazy barrier to entry to try these things out.
With that, I want to thank you again, Liam, and thanks all the attendees for participating.
Thanks, guys. Thank you, Thomas.
Thank you. Bye-bye.
Bye.